All right, so I'll just mention briefly that this kind of analysis that we just did with short read sequencing can also be done using the microarray technology that we talked about last time. The basic idea is, you'll recall, a microarray is a chip whose surface has spots of individual DNA probes, and this is one case where the probes are sensitive enough to see the difference of a single letter. Basically, the probes are quite short. So if a single letter doesn't match, the affinity of the DNA to bind to that probe goes way substantially down. So these DNA microarrays can distinguish a single letter difference. So typically what you would have for a particular SNP is you'd have one probe sequence that's designed to match the A allele and one probe sequence that's designed to match the C allele. You have your DNA sample that you're going to now put on top of the chip. That DNA sample is fluorescently labeled. So basically, wherever the DNA sticks, you'll see fluorescence at that spot. So if that DNA sample just had A, we'd expect the probe that has the A to be a bright spot and the C probe spot to be much lower fluorescence, versus the case where maybe half of the DNA has the C. Now we might expect an equal fluorescence between those two spots, etc. Okay? So is the technical readout of how the microarray reads out a SNP, is that clear to you? Any questions about that? So what the microarray mainly does is it allows you to do that assay in parallel on a massive scale. So millions and millions of SNPs on a little array about that big, that you can inexpensively assay all those SNPs in many, many, many samples. How large? How many probes? So a typical SNP chip could have, sufficient probes for all the common SNPs, say in the human genome, I believe. Say something like 3 million SNPs. So, how long are the probes? Typically in the neighborhood of 20 letters in length. So a single letter out of 20 has a substantial effect on the binding of the DNA to the probe. Okay, so just based upon the fact that people have 2 copies of each chromosome, we already said that the possible copy numbers are 0, 1, or 2, which turns into saying that 0% of the DNA would have a particular polymorphism, or 50%, or 100%. And so you're basically trying to bin the signal that you get for any probe pair into one of those 3 categories. Okay. All right. So this then we can distinguish different disease models. Sorry, question.
I just want to clarify that microarray chip does need to be configured to read the particular SNPs?
Yes, this is a crucial point. So the design of the microarray is the set of sequences that are used as probes. So you have to know beforehand what SNPs you're looking for in order to design that set of probe sequences that you then manufacture arrays with that particular probe set. So it's a one-time design and then manufacture a huge number of chips that would be used by many, many, many customers. There are alternate technologies that can do one-off microarrays, obviously more expensive, but feasible. So there's different kinds of microarray technologies.
How much would these chips cost?
I don't do the experiments myself, and it's been quite a while since I worried about the prices of them, but the different technologies, let's say, range in like $50 for a very cheap kind of microarray, let's say on up to several $100s. So that's the order of magnitude. Okay, so I'll just talk briefly about different inheritance models directly related to what we just said. This whole idea of there being 2 copies of each chromosome, therefore the number of copies you can have of some mutation, 0, 1, or 2. And in the so-called Mendelian inheritance model, that reads out as an actual phenotype, in other words, an effect on the actual characteristics of the organism, such as a disease, in the following way. Typically, and this is what's referred to as the Mendelian recessive model, we would say maybe we have one version of a particular gene that we would call unmutated, normal. I'm going to refer to that just as a plus. And let's imagine there's a mutation that does something bad to the function of that gene. So I'm just going to call that minus. You could think of that as being a loss of function. Maybe the gene doesn't work right anymore, doesn't work at all, or some aspect of its function is no longer working. So one of the reasons why we have 2 copies is it protects us from that situation. In other words, if you have one normal copy and one bad copy, which here I'm symbolizing plus minus, typically the result is no symptoms. You look normal. Okay? Only if you get minus minus, 2 bad copies, one from each parent, will you actually show disease symptoms. Okay? So this is the classic so-called Mendelian recessive model for a genetic disease, simple genetic disease. So obviously this is exactly like flipping 2 coins in a row and asking whether you get heads twice, right? If you're starting from parents who both do not have the disease but are carriers of the disease, in other words, they're both copy number 1 plus minus, okay? What's the probability if they have a child that child will end up minus minus and actually show disease symptoms? It's just 25%. Okay? Any questions? So an indominant mutation then, first of all, one of the parents would actually have to show disease, right? Because if they have any copy number other than 0, they're going to show the disease. And basically, ordinarily that would mean that they have copy number 1, because usually these disease mutations are quite rare. So to get 1 is rare, to get 2 is vanishingly rare, which would mean the child then would have a 50% chance of getting the bad copy from that parent. And then, since all it takes is 1, then they'd have the disease. Other questions? Okay. So I want to contrast that with another form of disease inheritance. You may be familiar with the fact that there is a special pair of chromosomes called X and Y in the human genome, and actually many other animal genomes. This is sort of the standard pattern in the vertebrates, where basically if you have 2 copies of the X chromosome, you're female. Whereas if you only have 1 copy of the X chromosome and you have 1 copy of the Y chromosome, you're male. So then from your parents, you simply receive 1 copy. So from a woman, randomly one of her 2 X chromosomes gets passed on to any child from her. And from a man, randomly one of those 2 X or Y chromosomes gets passed on to the child. So right away you can see it's a 50% probability approximately that you get Y versus X from your father. turning you either into a daughter or a son. Okay? So this clearly creates a special situation for men because they're only got one copy of the X chromosome. It means they're much more likely to show disease for any kind of disease, genetic disease, that's located in a gene on the X chromosome. Right? So all they have to get is one bad copy, and bang, they've got disease. So actually, there's a famous example of this, so-called hemophilia, failure of blood clotting. You get cut, just continues to bleed, 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 doesn't clot.